Okay, so last week I spoke about how the immune system is affecting with inflammation and everything, about how the immune system causes inflammation and that contributes to cancer. So today I'm going to talk about the immune cells that actually affects the cancer. The tumors. So macrophages functions by maintaining tissue homeostasis and repair. Activated macrophages, there's two types. There's the M1 type and the M2 type. The M1 type is pro-inflammatory, which adds to the tumors, and the M2 type is anti-inflammatory, which is anti-tumor. So macrophages that are anti-tumor displays an M1 polarization, which removes more immunogenic cancer cells. However, as the tumor progresses, the TAM elicits an M2-like polarization of TAMs, which is pro-tumor. So the promotion of the tumor by TAMs is stimulated by angiogenesis and lymphanogenesis of cancer cell proliferation and epithelial mesenchymal transition, thus also limiting the efficacy of the therapies. Um, so they can also function by opposing the tumor. They mediate the efficacy of the anti-tumor and anti-metastatic tumor of the histone deacetylase inhibitor. Another type of innate immune system cells is the neutrophils. These are the first cells recruited by damaged tissue. They remove pathogens by phagocytosis, secretion of antibacterial proteins, deposits of neutrophil extracellular traps, and exocytosis of protease containing granules. They function similar to macrophages. They also have two polarizations states, N1, which is pro-tumor. This aids the cancer cells, and N2, which is anti-tumor, which destroys the cancer cells. The recruitment of these cells are done by CECR2 ligands, which are secreted by the cancer cells. Okay. Another innate cells are the NK cells, also known as killer cells. The NK cells are effector cells with tumoricidal activity. They lack the receptor to detect antigens. However, they can recognize normal cells that are infected with viruses or tumor cells, such as the cancer. Their tumoricidal activity is general and non-specific because they are not induced by a specific antigen. This is opposed to T cells, T specific antigen cells. Studies suggest that the class 1 MHC molecules that are found on the surface of the normal cells inhibit the NK cells and prevent lysis. So this in turn means that the inhibitory receptors that target the cancer cells, which the cancer cells that lacks this molecule, allows the activation of the NK cells, and that's how the tumor lyses. So cells mediate the tumor killing by releasing cytotoxic perforin and granzyme, thereby eliminating tumor cells and triggering apoptotic pathways in tumor cells. This is another form of innate cells. They are found in every tissue throughout the body. They are dedicated antigen-presenting cells, which is opposed to the NK cells. They are found in tissues that are barriers, such as the skin, the lymph nodes, etc. They inhibit the tumor-specific immune responses. They are able to take up tumor-associated proteins, process them, and present TAAs to T cells. This in turn stimulates the CTL response against the tumor. DCs play a critical role in priming and consolidation of anti-tumor adaptive immune responses. Some classes of dendritic cells, which is also DC cells, can mediate tumor progression. A selective depletion of the CD141 plus DCs correlates with impaired NK and T cell activity, which thereby favors tumor progression. Tumors also secrete prostaglandin E2 to impair the DC's recruitment to the TME. T cells. This is a and this wait. Okay. T cells, they are normally found throughout the body to help prevent the autoimmune reactions. They are produced during they are the acquired immune system response. They are produced during the active phase of immune responses to limit the immune responses that may in turn damage the host. When these cells accumulate in cancers, they inhibit anti-tumor immune responses. During the early stages of tumor development, naive T cells produced in the lymph nodes migrate to the TME and mount a protective effector immune response, thereby eliminating the cancer cells. That's how they destroy them. They mount onto them and they shrink them. So high levels of the T cell infiltration results in a favorable prognosis. CD8 plus T cells differentiate into T lymphocytes and exert an anti-tumoral attack, which thereby results in the destruction of the cancer cells. Okay, the invariant NKT cells, which is a subset of T cells, they recognize lipid antigens. They have the potential to activate the absence of TCR simulation, and upon activate they secrete cytokines. These cytokines also are used to release the macrophages. These cells are not really studied that much, and they actually understudied compared to the other cells. B cells, these cells promote and support tumor growth. The lack of these cells, however, in studies show that the lack of these cells decreases tumor progression. So, however, the adaptive transfer of B cells restores the chronic inflammation, angiogenesis, and tumor growth. B cells also indirectly stimulate angiogenesis and chronic inflammation by activating myeloid cells. So normally these cells actually they kind of interlinked with each other. Um, oh, okay, let's just see there. There's an interlink between all these immune cells within the TME, suggests that during the phase of elimination, NK cells exert a strong tumoricidal role. That is the secretion of the CCL5, and this promotes the recruitment of the DC cells. The DC cells go to the TME. This results in the priming of the T cells, which in turn activates an immune response. So all these cells are interlinked. However, sometimes the cancer cells can escape the immune responses. They do this in two ways. In the first way, the cancer cells lose the expression of the tumor antigens on the surface of the cell. This in turn allows it to avoid being recognized by the cytotoxic T cells. The second way in which they can evade the immune cells is by instigating an immune tolerant TME. This happens when suppressive molecules such as the IL-10 and the TGF beta are produced. Along with them, the expression of the inhibitory molecules and the induction of the recruitment of the TMAs and MDSCs. This provides an efficient system for the for the cancer cells to evade the immune system.